Okay, everyone. I wanted to make sure we were really clear on how a neuron fired. So, when a neuron fires, that's called action potential. Okay, but as we talked about in class, before a neuron can go into action potential, it has to be at rest. And we have to understand what it's doing at rest to cause it to fire. So first we're going to draw our neuron. And remember, the signal in a neuron only goes one direction. It goes from the dendrites to the axon. All right, so you're going to have many dendrites on one side, and then the bulk here is the cell body that will have the nucleus in the middle. Coming down here is your very long axon, and it's going to end in what we call terminal buttons down here. We talked about that when two neurons interact, there's actually some empty space between the dendrites of one neuron and the axon of another. And we call that empty space the synaptic space. All right, so just like any other animal cell, the cell is surrounded by a cell membrane that's semi-permeable, meaning it lets some things in but not others. Okay, so we talked about two different ways that the cell membrane can let things in or out. One is a channel. And a channel, we said, was like that open door where things can go in and out freely. The other type is a pump. A pump has to use energy to pump things against the grain, against equilibrium. All right? We talked about three different types of ions that are at play when a neuron is at rest. So one is going to be a sodium ion. One is going to be a potassium ion, and those are both positively charged. And then the last one is going to be a chlorine ion. That one's going to be negatively charged. Okay, so at rest, the inside of the neuron, or the intracellular matrix, is mostly negative. Okay, we said that it has a charge of negative 70 millivolts. Okay, and that's because it has a pretty large ratio of negative chlorine ions on the inside. I'm going to color those in green so you can see them. Those negatively charged particles. Okay. Now, the special pump that are part of neurons are the sodium-potassium pumps. And what they're doing is they're pumping out positive sodium ions, and they're pumping in positive potassium ions. Okay, but we said that it does that at a ratio of 3 to 2. So for every 3 sodium ions it sends out, it lets 2 potassium ions in. I'm going to color those in orange so that you can see. So at rest, there's a high concentration of positive sodium ions in the extracellular matrix. So all of the juice that's around the cell. And it's mostly positive because those sodium ions are positively charged. And because the pumps are constantly pumping out sodium, we have a relatively low amount of sodium within the cell. So even though the sodium-potassium pump is letting in some potassium, right, which is still positively charged, it's outweighed by the large number of chlorine ions, which is why that charge on the inside of a cell is negative. The other reason is that the pump is pumping in the potassium ions, but some potassium ions can just leave out of these open channels. Just like every other thing in nature, things like to be at equilibrium. And so the potassium wants to go outside of the cell into the more positive environment. And so it'll just leave out of those channels. So this is this neuron at rest. And it has a lot of potential energy. And we can remember that because of action potential. It has the potential to want to change and get back to equilibrium. The first reason is that the charge wants to be at equilibrium. Because the inside is very negatively charged and the outside is very positively charged, the cell wants to be at equilibrium where the solution on the outside and the solution on the inside is about equal. Okay? So the charge is different across that cell membrane. The other thing that's different is the distribution of these ions. There's a lot of sodium on the outside, and because it wants to reach equilibrium, the sodium wants to be able to travel to the inside of the cell. There's not very much potassium within the cell, and so potassium naturally wants to travel in to have about an equal distribution of potassium as well. So there's a difference in charge, but there's also a difference in the distribution of ions. So there's two different things giving it quite a lot of potential energy. And the pump is keeping it at this tension. So what happens when a cell charges? Well, it reaches that equilibrium point, but it has to start with a trigger. It's not going to happen on its own. So what happens is we have these chemicals called neurotransmitters. Sometimes neurotransmitters are excitatory, meaning they make something happen. And sometimes the neurotransmitters are inhibitory, meaning that they stop a signal. So when an excitatory neurotransmitter crosses over from another neuron, it crosses over that junction, and it comes to the dendrite of the neuron. Neurotransmitters are really neat. They act like a lock and key system. So there's specific ports on the dendrites that accept specific neurotransmitters. So an excitatory neurotransmitter is going to interact with this lock and key system at the dendrite. And that acts as a catalyst to start the action potential. As soon as this neurotransmitter attaches to the dendrite, then we get a depolarization of the cell. All that means is that where the inside of the cell was once negative, it's going to become equal to the outside charge of the cell, and the charge is going to go from a negative charge to a neutral charge to briefly a positive charge. Okay, just like the extracellular matrix. Whenever this excitatory neurotransmitter hits the dendrite, then all of these channels start to open like little doors. It's an all or nothing response. So once a neuron hits action potential, it can't stop halfway. It has to go all the way through. And it only goes one direction. It's going to go left to right from the dendrites all the way down through the axon. So these doors systematically open all the way down the cell. And when these channels or doors open, then the cell is allowed to briefly go to equilibrium. The charges, so for example, the sodium will float into the cell, causing the negative intracellular matrix to become more and more positive until it becomes neutral. And the distribution of ions will change as well, where there's a more or less equal distribution of sodium, potassium, and chloride. As soon as these doors open, we get this charge that runs through the cell, kind of like a battery. And this action potential travels all the way down to the end point of the axons, the terminal buttons. Now, as soon as these little channels open, they're open very briefly and then they close back up so that the cell can return to a resting state. So it happens very quickly. And if you'll remember, part of what makes a neuron fire very quickly and able to communicate so fast with other neurons is because of these structures called the myelin sheath. Myelin sheath is white and shiny, and it wraps around the axon, kind of like in electrical wiring, where you would have the wires wrapped. That helps the signal to move more quickly. And so the signal hops all the way to the end. And then what happens when that depolarization or that change of charge reaches the end of the cell? Well, that causes a neurotransmitter on the terminal button to be released. Okay? That neurotransmitter, depending on the type of signal that was received, can either be excitatory, meaning it makes something happen, or inhibitory, meaning it stops the message. But if it's excitatory, it's going to travel through that synaptic space to the dendrite of the next cell, and the process starts over. And this cell will go from a resting state to an action potential, where all of that potential energy gets turned into an electrical charge, a depolarization that moves through the entire body of the cell until it reaches the axon on the other end.